Good morning and good day to each and everyone present here. It is my pleasure to welcome you all to a guest lecture on an overview of advances in the standardization of herbal drugs, organized by the Association of Pharmaceutical Research and by scientific partner BioLeaks Worldwide and by research partner United Innovators. I believe that video was informative to everyone here. On that note, I would like to take this opportunity to welcome our speaker for the day, Dr. Rokeya Sultana, Professor, Department of Pharmacognosy, Enepoya Pharmacy College and Research Center, Enepoya, deemed to be an university, India. Doctor, over to you.
Good afternoon, everyone. Myself, Dr. Rokeya Sultana, Professor and HOD, Department of Pharmacognosy, Enepoya Pharmacy College and Research Center. I would like to thank the organizer for giving me an opportunity to give a talk on an overview of advances in the standardization of herbal drugs. My topic includes what are herbal drugs and natural products, different facts about natural products, why natural products, herbal formulations, standardization of herbal drugs, an overview on advancement in standardization, natural products in drug discovery, their advantages, challenges, and how the recent advancement helps to combat these challenges, different opportunities. Let me tell you what are natural products. Natural products are the products or the drug or medicinal substances which are obtained from the natural resources like plants, animals, microorganisms, higher order animals, lower order animals, and minerals. So the drugs or the medicines which are derived from the plants are called as herbal drugs. So herbal drugs can be used as supplements or as a products like tablets, capsules, powders for the improvement of health and well-being of the humans. Why natural products? Natural products, if you look back the history of it, we can see since the ancient times, the people are using the natural product as the folklore medicine for different treatment of diseases and illness. Even though the drug discovery from natural sources are going through renaissance under the shadow of synthetic molecules. But new era has come with new possibilities where the novel targets as well as the state-of-the-art technologies can be implemented for the discovery of natural products. Let me tell you about some facts about natural products. Natural products offers uh, advantages as well as challenges during the drug discovery process in comparison with the conventional synthetic drug molecules. Natural products are having complex structure, which can be advantageous for studying the structural activity relationship where the synthetic routes are difficult. Even though there are so many natural products are available in the market, but the strong evidence regarding their safety and efficacy is not existing. I want to tell you about the fact that more than 50% of the FDA approved drugs, either their natural products or their natural product derivatives. Natural products is having a very interesting fact that it is having a selectivity to cellular targets. As a result, it can provide the selective targets for the disease-related ligands, as well as it can influence the disease-related pathways. So, it is easy for the natural product to shift the biological network from disease state to healthy state very easily. So herbal formulations are the products or the dosage forms which we get from the herbal drugs by implementing several authentic analytical methods which we can get a detailed profile of the phytochemical composition, marker compound present, as well as the nature about their major constituents. But it's a very major challenges to the researchers because there are so many constraints or the disadvantages present in quality control of herbal drugs. So the different constraints what we need to face when we are doing the quality control of the herbal drugs are the first of all, we have to mainly depending on the oil source because the wild source are giving the drugs. The natural products are not easily cultivated in all the weather conditions as well as environmental conditions. So, for the source of the drugs, we are depending on the wild source, which is not very easy steps to get the natural products. There are lacking in the information present in the herbal pharmacopoeias. Not all the natural products are well documented, and there are lack of public test house, lack of public safety evaluation facility, poor investment in R&D for herbal medicinal products, as well as there are lack of people who are trained with the traditional medicines. Also, the pharmacovigilance data are not collected. Like when we take the natural products as a medicines, we don't collect the data how safety the drugs are. And certification for the raw materials is also difficult to get. So very less number of certificates we can get. As a result, the raw materials are full of adulteration or substitution. As a result, there is lot of problem regarding the regulatory aspects of the natural products. So to combat all these challenges, a method called standardization can be implemented. So standardization is a very important step for the establishment of a consistent biological activity with a consistent chemical profile, which can simplify the product with a quality assurance program and for giving the product of the natural product with a quality production and manufacturing of herbal drugs. So there is a specific guideline given by the WHO for assessment of safety, efficacy, and quality of the herbal drug, which can reach the herbal into the global market. So if you are able to follow the guidelines given by WHO to achieve the quality, safety, and efficacy, the drugs can be easily accessible and available to the consumers, which is safe and effective in use. Let us see the overview of different techniques which can be implemented to overcome certain challenges. Various techniques such as extraction and characterization can be studied by using standard technologies. Nanomedicine standardization can be done. Different targeted drug delivery systems such as phytosomes can be used to increase the bioavailability. Different traditional medicines like plasma can be used as a nano carrier There are different metabolic methods or different advanced method which is called as metabolomics can be used for the development of phytotherapeutic agents to distinguish between the adulterants and discriminating the adulterants. We have the techniques called DNA-based molecular marker techniques and SCART markers techniques. We can also take help of different technology assisted methods for getting the high yielded herbal products like microwave assisted extraction technique, supercritical phase extraction technology, etc. Standardization also can be improved by taking help of advanced technologies like spectrophotometric method, chromatographic method, thermogravimetric method, capillary electrophoresis, polarographic techniques, and there is a technology which is called as nanotechnology, which can also improve the solubility and bioavailability of the herbal products. Herbal products also has to undergo different phases when it comes to the drug discovery and development. Different phases are drug discovery as well as screening of the drugs, lead optimization, uh, absorption, distribution, metabolism, expression and toxicity, clinical trials, NDA approvals, and new drugs to the market. This method has to be followed, whether it is a single drug or a new chemical entity or a biological therapeutics. So natural products used in the drug discovery are having several advantages and challenges, and which can be combated by using the different technologies. Advantages, as I have told, the natural product is having a very complex structure. So structure complexity can also bring certain advantages and disadvantages. Because of the complexity of the structure, most of the herbal compounds are having high molecular mass. The carbon atom and oxygen atoms are in sp3 hybridization mode. There are other points also which will helpful to study the structure in details in compared to the synthetic compound libraries. So these advantages can make the herbal products high rigidity for protein-protein interactions, and which will be also helpful for curing the different disorders. Natural products can be used as a lead compound and can also helpful for regulation of different defense mechanism, and it also can be used for infectious disorders. Furthermore, the traditional use can give a insight regarding safety and efficacy. Overall, the natural product pool can be used by enriching the bioactive compounds, and which will cover a wider area of chemical space compared with typical synthetic small molecule libraries. There are several challenges, but I have already discussed. To name few are: one is the adulteration, second was lack of standardization, third one is technical barriers to screen, isolate and characterize as well as optimize the product. So because of these problems, the pharmaceutical industries are declining in this interest for discovery of new drugs from the natural products. Let me tell you about what is the position of herbal drugs. The herbal drugs used in the natural product discovery is growing interest day by day. WHO has given the slogan "Health for All," and by implementing the prevention is better than cure, a lot of research are going on the natural product. As a result, multidisciplinary result has led to many new drugs as well as prototype active molecules and biological tools has come into the picture with the efforts of WHO. I can name few drugs which are used as a leading herbal lead for different disorders like hypertension, diabetes, cancer, dengue, immunomodulation, malaria, Parkinson disorders, etc. So these are the different drugs which are used for herbal lead. Let me tell you about practical outlook of the herbal drug discovery. How the herbal drug discovery is happening for the natural products from the natural products. So I want to tell you first, whenever we want to study any medicinal plants, the selection of medicinal plants and which pharmacological activity need to be studied, it should be very, very clear to the researchers. So there are 5 principles which are used to select the medicinal plants. They are random approach, taxonomical approach, phytochemical approach, ethnomedicinal approach, and information managed approach. So in case of random approach, what is happening? We will be selecting the medicinal plants based on the random selection. So we'll be collecting all the available plants in the area and will be identifying only on the basis of visualization and observation without having prior knowledge or experience about the selected plant. So it's a blind selection. Next is taxonomical approach. In this case, we'll be using the prior knowledge about the plant and interest about the genus and family of the plants. But here, not much knowledge about the phytoconstituents. Whereas the phytochemical approach, which is also called as chemotaxonomical approach, where both the knowledge of the bioactive chemical compound as well as the plant will be used. So, which is more interested approach. Other approaches are ethnomedical approach as well as information managed approach. In ethnomedical approach, what we are doing, we are using the information on the medicinal use of that particular plants in various areas. And lastly, the information managed approach, we are collecting the plant information based on the survey and use of the plants from the local areas, which will give the prior idea about their usage and activity, then their evaluation scientifically. So this is what about your plant material. So it is very, very important to select proper plant material. Otherwise, the entire research will be baseless. So once plant material is selected, the next will be extraction. So extraction is the process to take out the phytoconstituents or active constituents from the plant material by means of different solvents. So once we get the plant extract, we can study the toxicology and then further we can separate to get a pure compound, which will be elucidate the structure and study the toxicology and which can be used as a lead for the discovery of the natural products. So we have seen in the important steps for successful completion of natural drug discoveries are extraction, identification, isolation, separation, purification, and characterization. So if you are able to do these steps properly without any problems, then we will be able to have a new phytoconstituents with good bioactivity. So this new isolated phytoconstituents can be used as a lead compound for designing of new analogs with either improved therapeutic activity with reducing toxicity. And we can convert these lead phytoconstituents into medicinally important drugs by using herbal drug discovery and herbal drug advanced techniques. And we can bring this natural product for the socioeconomic benefits. But these steps for the biochemical and biological investigations of novel bioactive natural products are not so easy. They are challenging and time consuming. There are different challenges in different methods and which are can be used to separate by using different techniques. So in this flowchart, I am going to explain about how the solution outline for the traditional bioactivity guided isolation in natural products. So I'm going to show you the three different marks, three different colors boxes. So you can see the purple color, which is the purple color boxes, which is showing the method of drug isolation, where the problems are depicted in pink color and the solution is depicted in green color. So the steps is first we are collecting the fruit drugs from the natural sources, including the organisms, and next is we are making it into extracts. The extracts can be converted into the fractions, which will be having phytoconstituent rich compounds in it. Under different methods of fractionation, we can get the single compounds, and all extracts, fractions, and single compounds can be evaluated for bioactivity, and we can go for the discovery of natural products. Now, each steps there are certain problems and which can be solved by using different solutions. So, number of problems is the organism specially cannot be cultured without their natural habitat, and the product what we taken out whose where it is produced, which will not be of interest what we want. So this can be solved by using the advanced technology, by implementing new methods for culturing, like in situ method of analysis. We can also implement these methods for expression of biosynthetic genes, which will be helping us to identify the root of synthesis of the product. The next problem is the natural products are known, but when we don't know about what are the properties, drug-like properties, and these products are present in very less amount, insufficient amount. So we can solve the problem by implementing different methods of data application. We also can use different methods with the standard technologies for extraction and pre-fraction of the extracts, which can solve our problems. And the last problem is we can have the natural products which are having molecular targets, but we don't know how to find out the methods, how to trace the pathways. So here also we can find out the molecular mechanism, molecular mode of actions, and how to elucidate the structure, and we can solve the problems. There is fluorescence-based light cell assays, where we can study the entire pathways of the organisms using these methods. We have benchtop spectrophotometric method, which is one of the advanced techniques where we can do two, three methods together, like side explosion chromatography, NMR, and we can study the details about the phytoconstituents and some of the problems related to the isolation and separation will be solved. There are certain approaches which can be implemented for the natural drug discovery. One of them is top-down approaches and bottom-up approaches. So what happens is top-down approaches, what we do is, in the beginning only, we are starting with the organisms where we don't know what particular gene is responsible for the production of the natural products. Whereas in the bottom-up approaches, what will happen is first only we'll be able to identify the genes, and then we'll be using different techniques for the manipulation of the genes, and which you will come to know about the activity, and then we can go for the source. So where in the beginning only we'll be knowing what is the main pathways for doing this discovery, so it will be easy to do. Next approach is reverse pharmacology. Reverse pharmacology is just what we are doing laboratory clinic, just opposite to that. It is clinic to laboratories. What we do in laboratories, we first do the techniques is starting of the raw materials, and then step by step we go for the activities. Whereas in the reverse pharmacologies, we are first finding out what is the gene responsible for it, and then we go for the activity. So this was the technique which was invented by Sir Ramnath Chopra, as well as Gananath Singh. And this method has given a very important technique for in regards to the safety and efficacy of the product. So here I am able to show you the reverse pharmacology has already reduced the time, and it has made more economic, as well as it has made a safe for the Ayurvedic and traditional system of medicines. One of the important methods or techniques, analytical profiling techniques, is called as metabolomics. So metabolomics is used for measuring and comparing the large number of metabolites which are present in the biological samples. Here, several advanced techniques like high throughput analytical chemistry and other data analysis methods are used. So these methods can give a snapshot of the physiology of the cells, and it can give several applications for pharmacology, drug discovery, toxicology, plant biotechnology, system biotechnology, enzyme discovery, microbial biotechnology, food technology, as well as for diagnosis of different disorders. It can also use as a biomarkers and risk determinations. Very important is the recent advance is artificial intelligence. So artificial intelligence can help us to identify the compounds and drugs and also can help to modify the drugs based on the disease, and it can be widely implemented for the natural product as drug discovery. One of such methods is in vitro modeling for the neurological disorders, which is used by using human-induced pluripotent stem cells. So, stem cells in vitro models can be used for generation of the cells which are functioning in the neurons. So, in this case, we are using the somatic cells like the peripheral blood cells or keratinocyte cells, which will be used as a source and which can be exfoliated to the renal cells and can be converted into functional neurons, and will be able to find out what are the activity can be done in the neurological disorders. So this flowchart can show the how the novel drug discovery in case of neurological disorders is done through the high throughput screening. So these drugs can be studied on the brain related drug discoveries. So till now I have told you the advantages and the challenges. So let us see what are the opportunities are there for the natural product drug discoveries. So the number one, we are talking about the plant species. We need to know that we know that India is full with flora and fauna. Lot of plant products are there. So in order to screen thousands of plant species, we need to have to go for so many bioassays. We need to make so many extracts. So there is a very need to build a natural product extract library. I'm happy to inform you that Nicholas Piramal India has already built up a plant extract library which containing 6,000 extracts prepared from 2,300 plant species from India. Even though I told drug discovery is going through a renaissance in the shadow of the synthetic drug molecules, but the compounds which we get from the natural products are occupying a much greater chemical space than the synthetic chemistry methods. So WHO has told health for all. So the opportunities, if we are looking to see that in compared to the modern medicine, traditional system of medicines can cure and can reach to the economic people for several major health problems like cancer, hypertension, diabetes, malaria, viral, dengue, chikungunya, and recently the coronavirus also. So there is a growing interest in herbal drug development with minimum side effects, and there are better opportunities to explore the medicinal and biological properties of previously inaccessible natural products. To establish its usefulness, it is mandatory to focus on visualization and identification of unused herbal plants over the world. So we need to look back to the history. We need to see the previous discoveries of the naturally occurring products, and we need to present a picture of the current status, and we have to provide insight into the future opportunities and challenges for the drug discovery of new drugs to treat disorders. To summarize, natural products contain complex chemical structures which is differ from the synthetic chemical moieties, and high technologies when we are implemented and we are able to do new drug discovery, and we can benefit the whole world. So the world is always gifted with nature, the man is gifted with brain. So let us make use of it to discover new entities that will be available to common people in economical rate, and will be happy to lead a good life on this earth. Thank you.
Thank you, Doctor, for sharing your wonderful knowledge with us. I'm sure it will be of great benefit to everyone present here. On that note, I would like to take this opportunity to offer my gratitude to this guest speaker, Dr. Rukhaya Sultana, for spending your time, effort, and energy and sharing your knowledge with us. I'd like to give my thanks to the Association of Pharmaceutical Research, scientific partner BioLeaks Worldwide, and research partner United Innovators for organizing this guest lecture. And I would like to give my thanks to the listeners for spending your time and effort listening to the guest lecture. I'm sure you've all had getting beneficial takeaways from this session. Please stay tuned with us to learn about the further webinars and conferences that are going to be held as well. Thank you. Have a wonderful day, everyone.